It's a great pleasure and a little bit of a surprise, though I think I was told about this earlier, that I should introduce Sebastian. And then I was given a little note here, but I think I can do better than that. We recruited Sebastian. He was one of the first persons I recruited as chair of the Department of Brain and Cognitive Sciences. Now, Sebastian had done his PhD in theoretical physics from Harvard and then had worked in Bell Labs with David Tank in neuroscience. And we were intrigued by two things. First, we have been talking about how there are several very smart people in this room. And certainly, you are supposed to be smart when we recruit you to MIT. But I would say Sebastian was a couple of standard deviations above the mean, if you know what I mean. And then we were impressed by his ideas. Sebastian had lots of ideas. Of course, he was working on something very detailed, how the eyes move, and how there are integrators in the brainstem that make the eyes move. But we knew there was more promise there than that. And to make a long story very short, over the next 14 years, that promise is now being realized in spades. Sebastian has done some of the most creative work in the field in terms of understanding brain wiring. He has brought to bear a wonderful range of techniques and technologies that are promising to shape our field. He has written a book that, this is the part I can read, that somebody in the New York Times or in the Wall Street Journal says is the best lay book on brain sciences I've ever read. And so he's a great communicator and a wonderful scientist, and having laid this burden on Sebastian, I welcome Sebastian.
Thanks a lot, Mrugangka, for that very flattering introduction. So indeed, Mrugangka did hire me at MIT, and I guess he feels like he didn't make a mistake. So I'm grateful to hear that. All right, so Mrugangka says that I'm really smart, but I guess over the years I grew convinced that I really wasn't smart enough for the brain, for understanding the brain, that we needed to have more data. And so as he pointed out, I've been, in the last 6 years, I've been involved in the problem of trying to get better neuroanatomical data from brains and creating better ways of analyzing this data. And so what I'm going to talk about today is how far we've come, and also how I envision that this technology will be applied to search for connectopathies. So these are the long hypothesized miswirings of the brain that may be associated with mental disorders. I'm also going to talk about the problem of crowdsourcing, or the method of crowdsourcing. We have so much data that it really defies ordinary methods of analysis, and so our solution these days, my lab, is very much oriented towards the idea of building an online platform where we can invite the entire world to help us to analyze our data. And so I hope that some of you will come away from this thinking about ways where you yourself or you might enlist others to take part in this effort. Okay, so connectopathy is a fancy kind of word. Here's kind of a lay question that I pose to people. Is it really true that, as is often said, autistic people are wired differently? You'll see this in the media all the time. So what's the evidence for this kind of claim? And I'll show you, here's a review paper by Eric Krechesny and Karen Pierce, why the frontal cortex in autism might be talking only to itself. So this is the idea of over-connectivity, maybe locally, and then a long-distance kind of under-connectivity between the frontal cortex and other areas. Now, here's the kind of evidence that backs up this claim. Here's a graph, a very interesting graph of the behavior of brain size as a function of age. And what you're seeing here is not absolute brain size, but relative to the average. And you'll see then that at birth, on average, autistic babies have slightly smaller heads and brains. In infancy, they have somewhat larger, on average, heads and brains. And then that difference gradually goes to zero as they approach adulthood. So here is a kind of evidence then for a structural difference in populations between autistic individuals and those of typical individuals. But I want to remind you guys that although MRI is a very advanced 20th century method, this kind of idea of explaining individual differences goes back to the 19th century. And so here's a picture of two great writers. I'm not sure you will know either of them. They're a little obscure today, Ivan Turgenev and Anatole France. And the fans of Anatole France were disturbed to learn upon his death and autopsy that his brain was only 1/2 the size of Ivan Turgenev's. In fact, his brain was about 1/3 smaller than that of a typical individual's brain. And this caused a great deal of consternation. There were papers, scientific papers written trying to figure out if there's anything that could explain Anatole France's genius. But in the end, his brain is still 1/2 the size of Turgenev's. And yet Anatole France was the one who won the Nobel Prize in Literature. So explanations based on brain size have not really been very useful for understanding individual differences between typical individuals. And it seems to me they are important to have, but they are not going to explain differences between autistic individuals and typical individuals. It seems unlikely. And so that's why people have tried to go beyond the size of brain and its regions. People have looked at the shapes of neurons. They've looked at dendritic branching patterns. They've tried to count synapses. And in the finest kinds of investigations, they looked at dendritic spine morphology. Sometimes in certain kinds of brain disorders, you can find the dendritic spines are different in their number. They may be thinner or longer or shorter, et cetera, et cetera. So there's many papers on these kinds of abnormalities, and I've just written them here from sort of low resolution to high resolution. Now, some people dismiss the idea of looking for neuroanatomical abnormalities altogether. They think that it's really not very fundamental. And one of the problems in general with pathology is how do you distinguish between cause, correlation, or consequence? And of course, I guess when people look at brain abnormalities, they look at physiology, they look at anatomy, and they look at molecules. And I would argue that one of the reasons we love genetic studies of autism is that in the case of genes are so far upstream we would regard them not as a consequence or a correlation, but somehow involved in the cause because they happen early. Your genes are given to you at the beginning of your life. On the other hand, physiology in some sense is more like a consequence of an underlying, behavior and physiology are sort of downstream. They're kind of a consequence of some underlying abnormality. And so I would argue that anatomy still is somehow interesting because it still may be somewhat more causal than physiology. And maybe it's a way we can link the genetic abnormalities that people see with the physiological abnormalities. So all three of these levels are important. And I think what we need in anatomy is not to dismiss it, but to actually have higher resolution methods of seeing anatomy. And in particular, I'm really interested in serial electron microscopy. So I want to know about how neurons are connected at the cellular level, not brain regions, but microcircuits. And so if you haven't seen these kinds of images before, here's a really high-tech deli slicer from the lab of Jeff Lichtman, along with a tape collecting device invented by Ken Hayworth. So you can see the brain is on a chuck right there, piece of mouse brain, it moves back and forth. There's a diamond knife on the edge of the blue tub. The slices, 30 to 50 nanometers thick, float off into the surface of the water inside the tub, and they're collected automatically on that conveyor belt, forming a plastic tape, which is kind of like a movie, every frame of which is a slice of mouse brain. You can image those with an electron microscope, and we'll just zoom in on one slice of this piece of mouse brain, which is mounted on a plastic tape on the silicon wafer. And we can zoom in by a factor of 100,000 times until we get to the resolution where we can see the wires of the brain, the branches of neurons. And that by itself is not enough, right? We need to have three-dimensional images. So if you take images of many slices and you stack them up together, then you can get a three-dimensional image. And if you've never seen images like this before, you may find it hard to see the branches until we start to color one of them in. So we're coloring in the branches, coloring in the cross-sections of a dendrite of a neuron, slice after slice. And you can see in green an axon, a little short stretch of an axon. This is just a 6-micron cube. And they're touching in two locations. They're synapses, and we can zoom into the interior of the green neuron and see evidence, molecular evidence, that this is a synapse because you can see the vesicles inside. And this tissue actually wasn't stained as well as we'd like, and so if you have really nicely stained, post-stained tissue, you'll see also the postsynaptic density. You can't see that as clearly right here. You see kind of a smudge. Okay, so that's the synapse, and inside that 6-micron cube, there's a lot of branches of neurons, a lot of pieces of branches of neurons. They're all packed up like this. And in fact, you can see it's very complex. The total length of wires in your brain is probably about millions of miles if you do the estimates. This little piece of brain took 250 hours for my German postdoc Daniel Berger to color in. And I can assure you that he's much faster at this task than you are. He works with, many people, many labs have said he's the best tracer in the world. He never seems to get tired. He has incredible German precision. He's really incredible. Now, we've used this as a training set to do machine learning, to make the artificial intelligence do some of this task. And but that's still not enough. The task, computers don't see as well as humans do, so we have to enlist humans to help us. And so that's why we've resorted now to crowdsourcing. So here is a website called Eyewire. It's the first example of what I call citizen neuroscience. It emulates other kinds of websites like Galaxy Zoo and Foldit, but in the service of neuroscience. And so I'll just show you that website. You might want to go and play on it a little bit tonight. Oh. Let's see if I've got a network connection.
Are you running this from a lab computer?
No, I'm not, but I had a network connection until I rebooted my computer. So I wanted to actually show you guys the website, but maybe it's not going to work. What's that? Let's see. It's just more fun if I show you the live website. Oh, here we go.
To go to like a broad search.
No, here we
like
go.
The Harvard terms. There you
Here
go.
we go. Okay, here we go. So here's the site, Eyewire. Here's the call, discover neuroscience, trace the wires of perception. Here's a retinal neuron that's being reconstructed by our online community. And we can log in. And we can see on the lower left-hand corner, there's 10 users online. I can chat with them. On the right-hand side, we can see the leaderboard. So you can see Blistish, Jamie XQ, Mario Mar, and you can see the national flags beside them, and you can see that this is an international community of amateur neuroscientists. Mario Mar, I've had a little bit of email contact with them. Mario Mar lives in Bari, Italy. So it's kind of fun getting to know these people. And you can see, two more people have come online. And if I click this, we can actually zoom in onto a tiny little cube of the retina. And so this is your little game. It's like a three-dimensional coloring book, the pages of which are images of the retina. So on the left-hand side, you see the cube of the retina. On the right-hand side, you see one slice through it. And through the interactive 3D graphics, we can move up and down that slice and see all that tangled up retinal wiring. And the AI has colored in part of a branch, and your task is just to go through and correct it if it made a mistake. Add something if it left it out, erase something if it shouldn't have colored there. Okay, so that's the platform right now. And so what are we actually trying to understand? So I just want to say a little bit about the science, because sometimes people say, well, you're just looking at structure. Structure is not going to tell you anything about function. And it's true that structure doesn't necessarily solve problems about function, but it definitely gives us clues. And so I'll tell you about kind of an emerging scientific story that we're interested in right now. Here's the old retina that you may be familiar with from textbooks. 5 different classes, broad classes of neurons, and the connections between these classes of neurons are shown on the left. Now, contemporary retinal researchers know that there's a lot more kinds of neurons because each of those 5 classes is divided into many cell types. The current estimates, people are now saying maybe 100 different cell types in the retina, and so you can see down at the bottom maybe over a dozen different types of ganglion cells in the retina. Now they're saying 24. Each of those ganglion cell types is thought to be the output of a different visual computation. And one classic computation that has been studied in the retina for over 50 years is that of direction selectivity, the fact that some neurons will respond to visual stimuli moving in one direction but not in the other. And in 2008, a completely new type of direction-selective ganglion cell was discovered in the laboratory of Joss Sains, and Marcus Meister's lab did some physiology on it. And so these people found that this cell, which expresses the cell adhesion protein JAM-B, this cell has asymmetric dendrites. So you can see that the dendrites of all these cells, this is a transgenic mouse in which the JAM-B expressing neurons are labeled, and so all the dendrites are pointing downwards, and all these cells prefer motion that's downward on the retina. That's already interesting, right? Because a structure, this asymmetric dendrite, may be correlated with this function, which is detecting downward motion. And so here's a reconstruction of one of these J cells from the electron microscopy. So based on this electron microscopic image, we found an anatomically identified J cell. And I found a tree on the internet, so you can see that there's an interesting resemblance. And so one thing that's emerging is that J cells may receive direct path inhibition from starburst amacrine cells. So the red cell on the lower right-hand corner is another kind of retinal neuron, a famous one called the starburst amacrine cell. And there's a little red dot, which you may be able to see, which is a putative synapse between that the starburst cell makes onto the J cell. And what's shown in purple right here is all the parts of the J cell dendrite that are distal to this synapse. So what's interesting is that most of the bipolar cell input to this J cell is distal to the starburst synapses onto the J cell, which tend to be close to the cell body. And so what this may be, what we may have found is some evidence for a shunting inhibition model of direction selectivity in the retina, which was hypothesized by Christoph Koch, Tommy Poggio, and Vincent Tora back in 1982. Doesn't prove it, but we have a structural kind of signature which suggests that might be what's going on. And I showed this to Tommy Poggio the other night, and then he looked at me and he said, well, you know, I'm realizing that if you want to be a theoretical neuroscientist, you'd better live a long time. Because it's 30 years later, and this is really, I think, the first kind of evidence, some of the first evidence that this shunting inhibition mechanism they hypothesized might be operative in the retina. And so I said, Tommy, I'll drink to your health tonight. Okay, so that's just showing you, that doesn't prove anything, but it's a clue. And I think it can guide further research on how this direction selectivity property works. And so on November 26th, we're gearing up for J-Day. We're going to kick off an Eyewire campaign to look for directional dependence in starburst amacrine cell J connections. So we know they're connected, but we don't know whether there's a dependence on the direction that the dendrite points. We're really starting now with the marketing and publicity. It's going to be through social media and blogs, newspapers and magazines. We're getting the Eyewire community to get organized, to get the word out that here's a scientific challenge, that if we all come together, we can actually solve this and get enough data to conclusively settle some of these questions. Here is an email from an Eyewire, a testimonial. I recently got this. I must say how much I'm loving Eyewire. I never imagined there would be ways I could actually make a tiny contribution to real scientific discovery. Eyewire has become an important part of my life, and I feel a sense of responsibility for it, as well as personally gaining greatly from it. Here are some high school students who came to visit us at MIT this summer. They were participating in an educational program where they learned about neuroscience, and they also, at the same time, participated in science by helping us reconstruct neurons. There's a fun aspect to this, too. If I show you the forum, this is how we've gotten to know some of our users. Here's some, let's see. See if I can find this. All right. This was a gentleman called Jose. Jose was one of the first people to get onto the discussion forum when we opened it up back in March. He said, hello, fellow Eyewire. He said that he plays with his 7-year-old daughter. So we learned, hey, it's a family activity. We never actually thought about that until he said that. And then he, oops. Oh, yes, here we go. It seems like he found a piece of a neuron that looked like an ice skater, and so he put it up on here, and we all laughed about it. And then later on, there was some user who followed up on that suggestion and said, hey, I found a piece of a neuron, and I give you the finger. So as you can see, these users were really having fun. And I have to say, I go to these faculty meetings, and I hear all my colleagues griping at each other about what's going on in our department, why we're not teaching properly, et cetera, et cetera. So a lot of complaining, and then I go to this website, and I get to meet people who are just really excited about doing science and having a good time. And I start to remember, hey, that's why I went into science too. So that's a little bit of balance in my life to keep myself sane. Jose lives in Spain, and in fact, he's been translating our website into Spanish because we're trying to internationalize it to really get people involved. Okay, so I just want to say a few words then about the plan. So Mrganka's Simon Center has given us a little bit of seed money now to go after the problem of connectopathies. And so we're trying to get started now on this project. I would really appreciate your input. So I'm taking crowdsourcing very literally. I'm an amateur in this field, and so I would love if the scientific plan could be crowdsourced, if you guys could help me figure out what's the best way to go about this. And so we're starting out with the Shank3 mouse model from Guoping's lab. It, as you know, shows repetitive grooming, social deficits. Because Shank3 is well expressed in the striatum, people think that would be a good place to look for neuropathology. And indeed, Guoping's lab has already found abnormalities in medium spiny neuron dendrites and spines. So we're focusing on this idea that corticostriatal connections might be somehow pathological in restricted repetitive behaviors like those seen in autism. And there's two kinds of theories in the literature. One of them is the direct-indirect imbalance idea. The idea that there are these two pathways, the direct pathway and the indirect pathway, and they go through the striatum, and somehow this ratio of these two pathways is different in the repetitive behaviors. So we're going to look at this ratio and compare it in the Shank3 mutant and wild-type mice. The other hypothesis is the extreme habit hypothesis, the idea that plasticity is somehow responsible for these repetitive behaviors. And here what we want to look at is, again, corticostriatal connections, but compare those onto active neurons versus inactive neurons, because if there's a Hebbian kind of mechanism, an activity-dependent plasticity mechanism, the plasticity should depend on activity. And we can figure out these direct and indirect pathways and active versus inactive neurons by using fluorescence imaging. So D1 and D2 receptors can be marked, and immediate early genes can be marked fluorescently as a proxy for activity. And then if we combine the light and electron microscopy, then we should be able to not only look at corticostriatal connections, but know whether the striatal neurons are part of the direct or indirect pathways or were recently activated as part of activity-dependent plasticity. So that's the plan. Now, in general, I think one issue, though, is that I worry about, as everyone else does, about mouse models versus human models. How could we actually, if we did find a neuropathology that was intriguing, how could we follow up in human patients? And one thing I've noticed from the literature is that dendritic spine abnormalities, autism, and epilepsy have been linked in a variety of syndromes: Rett's, tuberous sclerosis, and Fragile X syndrome. And so I've been starting a collaboration with Sid Cash at Mass General Hospital. Here's an electron micrograph of a piece of temporal cortex removed during epilepsy surgery on a human patient. And so maybe in the sort of convergence of epilepsy and autism, we may be able to both combine human studies with mouse model studies. So we need your help. I would love the scientific community's advice on which animal model, what patient group, what neural circuit should we be studying, how should we go about studying it. Patient advocacy groups, a number of, I've actually went to the annual conference of the Phelan-McDermid Syndrome Foundation. They're really excited about the prospect of dedicating their own time to scientific research, to research on this disorder. And I think a lot of autism groups I've talked with are really excited about this idea, a new way of getting involved. Schools, as I've mentioned before. Corporate sponsors, so I think I'm actually trying to still negotiate with a certain corporate sponsors in the high-tech field because I want to finance this like public TV. So if you're a corporate sponsor, you get your logo on there, you get known as somebody who sponsors science, and lots of people will see it. So it's a win-win situation, and maybe in the pharma community, maybe in pharma companies or some other entities like that might want to have this kind of corporate sponsorship. I haven't explored that industrial sector yet. So I welcome all of your suggestions, and I'd like to thank my collaborators, Winfried Denk's laboratory in Heidelberg, Germany, Jeff Lichtman's laboratory at Harvard, and Richard Axel's lab. I didn't have a chance to talk about our work on plasticity with him, trying to find, that's actually our next project. It's going to be called pastsense.org, and so it's the challenge to see the memory of a smell, to see the structural changes that occur in the mouse piriform cortex caused by odor learning. Okay, and a number of present former lab members and the Eyewire community. And I'd like to refer you to my book where I explain the whole rationale for the search for connectopathies. Thanks a lot.